and welcome to Spotlight on Antibodies, a podcast from Biointron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in April, top news includes Fc engineering, FcRn inhibitors, and Tregitopes. Let's dive right into it. First up, Fc domain engineering for therapeutic development. Over the past 2 decades, the therapeutic potential of monoclonal antibodies has expanded dramatically. And this expansion isn't just because we're finding new biological targets. It's also being driven by remarkable advances in antibody engineering. Among these, engineering of the fragment crystallizable domain, or Fc domain, has emerged as a powerful strategy to modulate important properties like antibody pharmacokinetics, tissue distribution, and immune effector functions. Initially, the Fc domain was mainly recognized for mediating immune responses, especially through interactions with Fc gamma receptors and the neonatal Fc receptor, or FcRn. Now, though, the Fc domain is a key focal point for optimizing antibody performance across a range of therapeutic needs. From enhancing half-life and boosting brain penetration to fine-tuning immune activation and even overcoming manufacturing hurdles, Fc domain modifications offer a versatile toolkit. A good example of this is seen with antibody-dependent cellular cytotoxicity, or ADCC, a major mechanism by which antibodies help clear target cells. Enhancing ADCC often relies on strengthening the antibody's interaction on immune effector cells. Traditionally, glycoengineering strategies like reducing fucosylation have been used to achieve this, but they can add differences in manufacturing. A recent study offered an alternative. Researchers introduced point mutations into the Fc domains of anti-RHD monoclonal antibodies, specifically BRAD3 and FOG1, to enhance ADCC. Impressively, these Fc-modified antibodies, expressed in standard CHO cell lines, preserved their RHD binding and maintained high production yields. Most importantly, they demonstrated stronger Fc gamma R3A engagement and ADCC activity, comparable to clinically used RHD PIGG, a treatment used to prevent hemolytic disease of the fetus and newborn. This supports the idea that Fc protein engineering can meaningfully enhance antibody function without needing to alter glycosylation patterns, a major manufacturing advantage. Moving on to another topic, FcRn binding modulation for CNS delivery. One of the biggest challenges in antibody therapeutics has been delivering biologics to the central nervous system due to the protective blood-brain barrier. FcRn is expressed on brain endothelial cells and mediates transcytosis of IgG, offering a possible transport pathway into the brain. A recent study from the University of Buffalo used human FcRn transgenic mice to explore this. They examined different trastuzumab variants engineered with varying FcRn binding profiles. Mutants that had increased FcRn binding at both neutral and acidic pH, specifically the YPY and YQA-QY variants, showed higher brain to plasma and interstitial fluid to plasma ratios compared to wild-type antibodies. Meanwhile, the YTE variant, which binds only at acidic pH, showed no increase in brain exposure. And antibodies like the IHH variant, which lacked FcRn binding, exhibited reduced brain uptake but longer ISF retention. The takeaway here is that enhancing FcRn binding at neutral pH is essential for efficiently transporting antibodies across the blood-brain barrier. This insight could be pivotal for improving CNS delivery of antibody-based therapies. Beyond delivery challenges, preclinical prediction of pharmacokinetics is another critical piece for advancing Fc-engineered antibodies. Human FcRn transgenic mice, specifically the TG32 strain, are commonly used for this purpose. Recent studies show that the Fc-engineered antibodies designed to bind to FcRn more strongly maintained low clearance in these models even when challenged with competing high-dose intravenous immunoglobulin. Wild-type antibodies, in contrast, exhibited increased clearance under the same conditions. Using allometric scaling, researchers could accurately predict human plasma concentration time profiles from the mouse data. This validates the use of FcRn transgenic mice for modeling the pharmacokinetics of Fc-engineered monoclonal antibodies, an important step for accelerating early-stage drug development. One last consideration in Fc work is aggregation. Forced aggregation of an IgG1 antibody showed increased binding to all Fc gamma receptors in certain surface plasmon resonance experiments, especially Fc gamma R2a. But when aggregation-sensitive formats weren't used, the effect disappeared. Functionally, Fc gamma R2a mediated activity slightly increased with aggregates, while Fc gamma R3a activity decreased, likely tied to glycosylation changes. The main takeaway: controlling aggregation during manufacturing is critical because it can significantly alter therapeutic behavior. Now, shifting gears, let's dive into FcRn inhibitors and the explosion of innovation happening there. Just last month, Merida Biosciences came out of stealth mode with a $121 million Series A raise. Their focus: developing precision therapeutics that selectively eliminate disease-causing antibodies, leaving the rest of the immune system untouched. Their platform uses novel Fc protein engineering to achieve deep, durable depletion of pathogenic antibodies, with initial programs targeting diseases like Graves' disease, primary membranous nephropathy, and severe allergies. In parallel, the broader field of FcRn antagonists is booming. Blocking FcRn prevents IgG recycling, leading to enhanced lysosomal degradation and lower IgG levels, a promising mechanism for treating autoimmune diseases like myasthenia gravis, immune thrombocytopenia, and chronic inflammatory demyelinating polyneuropathy. A recent clinical review shows that FcRn inhibitors are highly effective at lowering IgG levels. But one caveat is emerging: hypoalbuminemia. Since FcRn also helps recycle albumin, blocking it can cause albumin loss. A mechanistic study from the University of Southampton found two drivers behind this side effect: enhanced degradation of FcRn itself, and competition between albumin and FcRn antagonists for binding. Understanding these dynamics is critical for designing the next generation of FcRn inhibitors that maintain specificity while minimizing metabolic disruptions. One example in the field is Johnson & Johnson's nipocalimab. This antibody binds a unique epitope on FcRn and works across both extracellular and intracellular pH conditions. Studies show that it can selectively deplete IgG without broadly affecting other parts of the immune system, making it a strong candidate for diseases involving autoantibodies and alloantibodies. Beyond autoimmune conditions, Fc engineering is also opening doors in infectious disease. In tuberculosis research, antibodies engineered to boost Fc effector function were shown to enhance neutrophil survival and activation, resulting in stronger pathogen control. This shows that antibody Fc design isn't just about neutralizing pathogens. It can actively modulate immune cell behavior to drive better disease outcomes. Finally, let's explore a fascinating topic: Tregitopes and immune tolerance. As antibody-based therapies expand, one major challenge is managing the body's immune response to these biologics. Problems like immunogenicity, loss of tolerance, and unintended inflammation can threaten both the safety and efficacy of antibody treatments. This is where Tregitopes, or regulatory T-cell epitopes, come into play. Tregitopes are short peptides that bind MHC class II molecules and activate natural regulatory T cells. They reduce inflammatory signals, calm down antigen-presenting cells, and expand populations of Tregs producing IL-10 and TGF-beta, all without globally suppressing the immune system. Preclinical models show that Tregitopes have promise in treating autoimmune diseases like Guillain-Barré syndrome and inflammatory bowel disease. They're also being explored for vaccine design, enzyme replacement therapies, and maternal fetal tolerance. For antibody developers, Tregitopes offer an exciting opportunity by incorporating them into antibody designs or delivering them alongside therapeutics. We may be able to reduce immunogenicity and extend antibody durability in patients. However, a recent study looking at somatic hypermutation, the process by which antibodies naturally evolve to better recognize their targets, found that Tregitope content tends to be lost as antibodies mature. The more an antibody undergoes SHM, the fewer Tregitopes it contains, and the more effector T-cell epitopes appear. This means that, ironically, natural affinity maturation can make antibodies more immunogenic over time. It highlights the need to screen for and reintroduce tolerogenic epitopes during antibody engineering, especially for chronic or high-dose therapies. Further adding to this picture, recent research revealed that antigen-specific Tregs, those matching the same peptides as pathogenic effector T cells, are essential for restraining autoimmunity. When Tregs share antigen specificity with harmful T-conv cells, they can suppress autoimmune responses much more effectively. This suggests that engaging Treg cells with matched specificity could improve antibody therapy outcomes, especially for self-antigen targeting. Finally, a promising universal approach is also being explored. Scientists have identified conserved Tregitopes from proteins like alpha-tubulin, which seems capable of inducing broad tolerance across different immune responses. These sequences mimic ancient parasite antigens and have shown the ability to suppress immune activation in a variety of settings, opening the door for designing tolerance scaffolds to accompany biologic therapies. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow Biointron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.